finish off chapter 17 notes, we're going to do this video, and then I'll give you some class time on Friday to work on the summary and learn smart that is due for this, because I only gave you this short of week when I usually give you 2 weeks. And so our lesson objective is going to be that you're going to be able to analyze data related to questions of speciation and extinction throughout the Earth's history and justify the selection of data that addresses questions related to productive isolation and speciation. And then lastly, also be able to describe speciation in an isolated population and connect it to a change in gene frequency, a change in the environment, and natural selection or genetic drift in action. And so the do now is that I'm going to ask that you define and distinguish each of the following types of species definitions. And so if you go to your textbook pages and you use a search function, you can actually search up morphological species or speciation, or you can search up phylogenetic speciation or evolutionary speciation or biological speciation. And you should be able to write out a brief answer and advantage and example of each. So I'm going to ask that you pause and go ahead and do that. Okay, so in summary, the morphological species formation theory or concept is that each species is defined by one or more distinct physical characteristic called a diagnostic trait that distinguishes the species from each other. So they're different from each other based on that. And morphology is usually talking about the anatomical or physical body of animals. And so the advantage of morphological species definitions is that you can define fossil species based on only those traits that are preserved in the fossil record. So it's kind of easy to see evidence of the species in how you group the species based on their fossils and their bone structure. So the examples on the right, where you can see that it's boxed and showing that there's bone structures in similarities in bone structures between humans, dogs, birds, and whales, and therefore you'd be able to not necessarily put them in the same species, but you'd be able to identify that they have similarities, so they might have a common ancestor, and that if you find, for example, 2 specimens of whale fossils, you'd be able to see that they are in the same species because their morphology is similar. Coming up next is phylogenetic species definitions, and so the concept is an evolutionary family tree or phylogeny is used to identify species based on a common ancestor, that is, a single ancestor for 2 or more different groups. And so the advantage of using this concept is that it does not rely on morphological traits to define a species. You can actually use other characteristics. You can use information from DNA and genetics as well in order to create your phylogenetic tree, as you see in the example on the right, where you can use traits like mammary glands, having live-bearing children, having a placenta, to then separate out different groups and get more and more specific as you use more and more characteristics until you can actually speciate every single type of organism you're grouping, for example. The next one we have is evolutionary species definitions, and the concept goes as a species is descended from a common ancestor and has its own distinct characteristics and behaviors. This one actually looks pretty similar to the phylogenetic one because it works in a sort of tree format as well, and there's going to be cladogenesis or anagenesis where traits are being developed or differences are being developed. And so then speciation happens, and what was originally once one species now branches into 2 different species, whether because they're changing from geographic isolation or reproductive isolation, as you can see in the example on the right. And the advantage of using the evolutionary species definition is that it is explainable and can help you explain the fossil record, because we have the record of what organisms look like throughout time as you go through the layer or strata of the rock below the Earth's surface. And the last species definition we're going to go through is the biological concept, and that one says that we can identify species based on whether 2 or more populations are reproductively isolated from each other. So if they are reproductively isolated, it means that they cannot have viable offspring together. So maybe they can have a baby, but the baby can't have more babies. Or they can't even make babies to begin with, either because they're physically incompatible, they are regionally never going to interact, or other things that are preventing them from ever having offspring. And the advantage of this system is that it's very black and white. Either you can have offspring that are viable or you cannot. And the examples would be closer related species like the carrion crow and the hooded crow can make hybrids, which means that they're closely related. However, these hybrids are sterile and cannot produce more offspring. And species that are not very closely related at all can't even make a baby to begin with, usually because their behaviors are too different, their routines that they would never interact with each other, even if they live in the same area, or they don't even live in the same geographic area. So these are some interesting species definitions. So we're going to read through a little bit of text, and then you're going to use this information to answer a short response. So the morphological species concept identifies species based on diagnostic traits. Actually, this is kind of already what I went through. Yeah, so this is from your textbook, actually. So if you're going through your textbook and you're going to be doing your Summary and Learn Smart, you'll actually see the section that kind of identifies some of the species concepts that we just talked about. And you can read through it here, or you can wait till you get through your summary. So how do new species evolve? There are two kinds of processes that will happen, and the main ones are pre-zygotic and post-zygotic. So these two are the main categories. Pre-zygotic, as you can tell from the pre, means before the zygote is formed or before the baby is going to even be formed. This is like before sperm and egg even meet. And post-zygotic would mean after the sperm and egg meet, whether the baby forms or whether the baby can even actually fully grow in the womb or in the egg, or even after the baby is a fully functioning adult, whether or not it can reproduce and continue on its lineage. So there are some isolating mechanisms that cause organisms to become different and eventually speciate into new species. So like the definition, it says pre-zygotic isolating mechanisms prevent mating attempts or make it unlikely that fertilization will be successful. So anything up until the point of sperm and egg meeting, these are some ways that are preventing species or organisms from making that happen. So habitat isolation would mean that species occupy different habitats, so they're never even going to encounter each other to mate. Temporal isolation, which means that they reproduce at different times. And this isn't saying like morning or night. This is usually saying different times of the year. It could be morning or night. It just depends on the routines or habits of the species. Behavioral isolation would be that courtship patterns are different. So mates actually can only recognize the courtship patterns, for example, bird calls or bird dancings or bee dancing for their own species. It's different for every species. And so that way we don't really get any interbreeding because they actually don't recognize the courtship of another species. Mechanical isolation would mean that the animal genitalia actually do not match together. Or for example, the plant floral structures, the pollen can't extend a tube long enough down every single flower's style. So then it's not going to be able to reach the ovules in the ovary, right? So different mechanisms set up for reproduction are incompatible with certain other ones. Sometimes there's broad category ones that will be compatible, but then the sperm and the egg actually just don't fertilize. And then gamete isolation is that gametes that meet do not fuse to become a zygote. And that's what I was just mentioning. So even if the sperm and egg do meet, if they're two different species, they still sometimes just can't even fertilize and become a zygote. So post-zygotic isolating mechanisms prevent hybrid offspring from developing or breeding. So here is hybrid inviability. So the hybrid zygote is not viable and dies. So the sperm and egg meet, fertilization happens, but then the hybrid zygote cannot survive and dies before it's even born. Hybrid sterility is the hybrid zygote develops. So the sperm and egg meet, they fertilize, they produce a zygote, and the zygote develops into an embryo, and the embryo develops into an individual organism that is a hybrid. However, this hybrid cannot reproduce with anybody. So here is kind of a visual representation of where it's happening or where each of these mechanisms are happening. So there's pre-mating, there's during mating, and there's also fertilization or post-fertilization, post-zygotic isolating mechanisms. So take a quick look over this one. So use the figure below to write a few sentences describing the process of speciation. So you're going to take a look at what's happening for branching and lineages, extinctions, microevolution, macroevolution, and describe how post-zygotic and pre-zygotic reproductive isolating mechanisms help facilitate this speciation. So take a pause and go ahead and do that. We are actually going to read through this section together, even though it is in your textbook, because it comes with the next part that you're going to do a quick short write about. So Hox genes have been found in all animals, and other shifts in the expression of these genes can explain why insects have just 6 legs and other arthropods, such as crayfish, have 10 legs. In general, the study of Hox genes has shown how animal diversity is due to variations in the expression of ancient genes, rather than to wholly new and different genes. So pelvic fin genes. The three-spined stickleback fish occurs in two forms in North American lakes. In the open waters of a lake, long pelvic spines help protect the stickleback from being eaten by large predators. But on the lake bottom, long pelvic spines are actually a disadvantage because dragonfly larvae seize and feed on young sticklebacks by grabbing them by their spines. The presence of short spines in bottom-dwelling fish can be traced to a reduction in the development of the pelvic fin bud in the embryo. And this reduction is due to the altered expression of a particular gene called PITX1. Hind limb reduction has occurred during the evolution of other vertebrates. The hind limbs become greatly reduced in size as whales and manatees evolved from land-dwelling ancestors into fully aquatic forms. Similarly, legless lizards have evolved many times. The stickleback study has shown how natural selection can lead to major skeletal changes in a relatively short time. For human evolution, the sequencing of genomes has shown us that our DNA-based sequence is very similar to that of chimpanzees, similar to mice, and indeed all vertebrates. The human genome has around 23,000 genes. Based on this knowledge and the work just described, investigators no longer expect to find new genes to account for the evolution of humans. Instead, they will predict the differential gene expression, new functions for old genes, or both will explain how humans evolved. We will discuss the details of human evolution in chapter 30. So as with all genes, mutations of developmental genes occurs by chance. And this is a random process that creates variation. Without variation, evolution cannot occur. Even though mutation is random, natural selection is not a random process. Rather, natural selection acts on the variation that is present in a way that favors the survival of advantageous traits in a particular environment at a particular time. This should not be misinterpreted as that evidence that evolution works towards an end goal or an optimum. Evolution is a perpetual process or continual process that leads, that shapes variation from generation to generation. Where this process leads is unpredictable and is dependent upon a complicated array of external forces. In the next section, we will observe that evolution is not directed towards any particular end. So macroevolution is not goal-oriented. And this is important, and I cannot stress it enough that people are like, well, you're just trying to make the optimum creature. And there's no such thing as an optimum creature in an area because there's always going to be variation and things are always changing in the environment to begin with. Right. So the first evolution of the Horus Aqueous has been studied since the 1870s. And at first, the ancestry of this genus seemed to represent a model for gradual, directed evolution towards the goal of the modern horse. There were three trends that were particularly evident during the evolution of the horse. So we saw an increase in overall size, we saw a toe reduction, and we saw a change in tooth size and shape. By now, however, many more fossils have been found, making it easier to tell that the evolutionary history of the horse is complicated by the presence of many lineages that actually evolved, went extinct, and were not on the lineage that led to the modern horse, right? So there were branches that went extinct, not directly tracing into what we currently have and know as horses. So if you look at the tree down below, it is an oversimplification because it's based only on the evidence from the few fossils that we have. And there are probably a lot more fossils that have to be discovered if we ever get to. So if the evolution of the horse were directed towards the goal of the modern horse, we would expect actually only one single branch on this tree with the intermediate fossils leading directly from the ancestor to our common horse. But it's actually an evolutionary tree that has many branches, and we will probably have even more added to it once they discover more fossils. So each of the ancestral species of the horse was adapted to its environment. Adaptation occurs only because the members of a population with an advantage are able to have more offspring than other members, right? Increased fitness. Natural selection is opportunistic. It's not goal-oriented. It's whoever at the current moment is the best able to survive is going to be able to best produce more offspring. Fossils named Hirachothirium have been designated as the first probable member of the horse family living about 57 million years ago. These animals had a wooded habitat, ate leaves and fruit, and were about the size of a dog, which is pretty different from our current horses. Their short legs and broad feet with several toes would have allowed them to scamper from thicket to thicket to avoid predators. Hirachothirium was obviously well adapted to its environment because this genus survived for 20 million years. The family tree of Aqueous does tell us more about that speciation, diversification, and extinction are common occurrences in the fossil record. The first adaptive radiation of horses occurred around 35 million years ago. The weather was becoming drier and grasses were evolving, so eating grass requires tougher teeth, and an increase in size and longer legs would have permitted greater speed to escape enemies. So the second adaptive radiation of horses occurred around the 15 million years ago mark and included Merychippus as a representative of those groups that were speedy grazers who lived on the open plain. By 10 million years ago, the horse family was quite diversified. Some species were large forest browsers, some were small forest browsers, and others were large plain grazers. Many species had 3 toes, but some had 1 strong toe, which is the hoof that we see nowadays. Modern horses evolved about 4 million years ago from ancestors who had features that are adaptive for living on an open plain, such as large size, long legs, hoofed feet, and strong teeth. The other groups of horses prevalent at the time became extinct, no doubt for complex reasons that we are still discovering to this day. Humans have corralled modern horses for various purposes, and this makes it difficult to realize that the traits of a modern horse are adaptive for living in a grassland environment, because we actually keep them in barns and we keep them in fields, which are kind of similar to the environment, but very controlled. So you're going to use evidence based on examples and reasoning to write a couple sentences explaining why we are not evolving towards some perfect organization, because evolution is not goal-oriented. You can use the examples shown because they're the ones showing the evolution of primates and eventually branched into humans, as you can see up here. So go ahead and pause and do that. There are some developmental genes in macroevolution that are the same genes, but they bring different changes in body shapes and organs by gene expression being different, or the developmental process that occurs after it's been expressed. So some examples are the PAX6 eye development gene, the TBX5 in limb development gene, and the Hox genes that we were just talking about for overall shape. For example, for PAX6, if there's eye development, it tells you what kind of eye you have, where it's going to be if the gene is being expressed in a certain area, and what type of vision is going to be best implemented. TBX5 gene, like we explained earlier, like the insects versus the crayfish or crawdads, they are, the expression of this gene tells you how many limbs you're going to have. So humans have 4, insects have 6. Some crustaceans have 10. So it depends on the expressions of these genes, even though the genes are all the same gene. Right? We actually explained this one in the last video lesson that was a while ago, where you got to see where wherever the PAX6 gene is expressed, it's going to grow an eyeball there. So even if it's expressed in the wrong place, like on the antenna of a fly, it's going to try to grow an eyeball there. Because the PAX6 gene is in every single cell, in every single location on every single part of your body, but it's not always expressed, right? Let's talk about autoploidy and alloploidy. So autoploidy and alloploidy are 2 things that happen in plants, usually and most commonly, because when plants have extra sets of chromosomes, it actually sometimes makes them stronger and better at surviving in environments. And most of the plants that we eat and cultivate for crops are actually ones that are autoploidy and have a lot of copies of chromosomes. And so autoploidy, for example, is when a diploid plant produces diploid gametes due to non-disjunction. So instead of splitting in half the number of chromosomes, they accidentally put their full number into their sperm and egg or their pollen. And so a diploid gamete, which is 2N, fuses with a haploid gamete, which is just N, and then you end up with this weird 3N plant. And the triploid 3N plant is steroid. Notice it's triploid, right? Because it's got 3 sets right now. It's sterile because the chromosomes cannot pair during meiosis. And then also, it doesn't have any other plant that is also 3N to even have, you know, pollen transfer with to produce offspring in the first place. Alloploidy is an interesting case where it requires 2 different but related species of plants. So you get hybridization and you get a combination of chromosomes from 2 different species. And it means that the offspring is viable. Oh, sorry, isn't viable. The offspring does survive, but it can't necessarily reproduce anymore with any future other organisms because its chromosomes don't match with anybody's chromosomes anymore. It's a random combination of 2 different species. So take a look at this diagram showing these 2 types of flowering plants. And tell me, is this autoploidy or is it alloploidy? And justify your answer. So pause and do that. I'm going to ask you to also quickly look at these 2 diagrams. So there's one diagram on the left that is a gradualistic model or gradualism for evolution that we explained earlier with the horses, right? One single line all the way up until from the ancestor all the way up until the current bird, where it's only species along that same line. And then there's also punctuated equilibrium, which has a bunch of different species and a bunch of different branches, and all of them are different, that stem from one common ancestor, and they're not all going towards this one target goal of today's current bird. So you want to take some time and describe what is gradualism and what is punctuated equilibrium using these examples. So pause and do that. So like I said before, let me know, is this gradualism or punctuated equilibrium? And then justify your answer based on the branching pattern. Is it a straight line or has it got many branches? Pause and do that. So we're going to talk about reproductive isolating mechanisms again, and we're going to go over a little bit in more detail of what they are. So this is again pulled from your textbook. There are physiological, behavioral, and genetic processes that inhibit interbreeding. And so if organisms cannot mate and produce offspring, or their offspring are sterile, that means that they've officially separated into two different species. So like I said before, there are pre-zygotic isolations and post-zygotic isolations, and it's just pre-fertilization or post-fertilization. Some pre-zygotic isolating mechanisms are habitat isolation, where two species occupy a different habitat, so they can interact. Temporal isolation, so if they reproduce, one reproduces during the spring, the other species reproduces during the fall. You'll never have to worry about their reproductive periods overlapping. Behavioral isolation, right? If males and females have courtship and they don't match up, right? You're not going to have a bird going after a male bird going after a female that doesn't present in the way that it thinks is attractive, right? Because they have different behaviors. Mechanical isolation means that physical incompatibility could be an issue. And then gamete isolation just means that again, sperm and egg, even if they meet, they won't fertilize. Post-zygotic in isolating mechanisms are like hybrids form from the fertilization of the sperm and egg, but they die in the womb. Or if it's sterility, the hybrid actually does form and is born and becomes a full adult and reaches sexual maturity, but cannot mate with anything because it doesn't have any similar genetics with anything else. So the summary, we have it down here. So mules are an example. If you made a horse with a donkey, you can get a mule. And they're hardy pack animals that are also good for traversing mountainous areas because they're not as big as a horse and they have the sturdiness of a donkey. But they're sterile because they can't reproduce with anything else because none of their genetic combination really matches even other mules. Over here, we see blue-footed boobies. They have a specific mating pattern where they dance around and smack their feet around like tap dancers, and it's really cute, but they're not really going to be attractive to, for example, a seagull, which mates, or an albatross, which meets, courts, and mates all in the air while it's flying long distance, right? So they all live in the same habitat area, but their behaviors are different. So that would be behavioral isolation. So define the following and give two examples. If you need to look up the two examples, you can go ahead and look them up. Two examples for pre-zygotic and two examples for post-zygotic isolation. Pause and go ahead. Okie dokie. So here is some information. I'm going to ask that if you want to read it, you can pause and go ahead and read it. You're going to encounter in your textbook, but it'll help you answer the stuff that we're about to come up on. So we're going to define some speciation mechanisms, and you're going to research / Google some examples of how they occur in nature. So allopatric speciation is a model that proposes that new species arise due to an interruption of gene flow between populations that are separated geographically. So usually when there's groups that have constant gene flow, they'll have mating and their gene pool is pretty mixed and similar. But once there's a separation and they're no longer mixing, they are adapting, they're becoming more and more suitable to their specific environments, right? And they're no longer interacting. So they're no longer getting the variations that occur in the other population. So they can become more and more different over time if their environments especially are different. Okay, so that is a model. So I want you to look up an allopatric speciation example of a plant and an animal. There are plenty of examples. There's another version that explains that sympatric speciation, which is the origin or new species and populations that overlap geographically. So you're going to look up plant example and an animal example for sympatric speciation. So the geographic barrier is not required, and speciation is simply a change in genotype that prevents successful reproduction. It could happen due to mutation, it could happen by random chance, it could happen by behavior changes or preference changes by animals too. It just is a way that new species develops, even if there isn't any physical geographical separation. Autoploidy, like I said before, is a polyploid organism. We said triploid before, but sometimes it can be 5s, 7s, 9s, it could be 8s even, it can be even numbers as well, as long as it doesn't match up with anybody else. Ends up being an awkward number where the plant is kind of stuck and not be able to reproduce. So Autoploidy would be a polyploid organism with multiple chromosome sets, all from the same species. And if there's too many sets, then it can't reproduce because it doesn't match with its own species anymore. So find me a plant and an animal example. And then alloploidy is a polyploid organism that contains the genomes of 2 or more different species. So this would essentially be like hybridization. So I want a plant and animal example of those. There's also reinforcement, which is the connection between natural selection and reproductive isolation that occurs when 2 closely related species come back into contact after a period of isolation. So this is related to species either being able to become different, and then when they come back together, it's kind of reinforcing that they're no longer the same species because they can't breed together. Or if they come back and they still can, that means either they haven't separated for long enough to actually become different species, and now they're going to intermingle, so they're back to square one. Adaptive radiation is the rapid evolution of several species from a common ancestor into new ecological or geographical zones. List a plant and animal example. And then convergent evolution is similarity in structure in distantly related groups, generally due to similarly selected pressures in the environment. So it means that if there's a need, natural selection and evolution will eventually provide a medium or a mode to meet that need. And in many environments, there are similar needs, but different organisms or different species are adapting to those needs. So for example, convergent evolution could be the ability to fly. And that is a trait that has been developed in many different organisms. It's been developed in birds, and it's been developed in bats, it's developed in insects, right? They all have a very different morphological, physical, structures, but they've converged in similarity because they all have a need to be able to fly for traveling, for catching food, and so they have similar structures in that respect. So give me some plant examples. Give me a plant example and an animal example of convergent evolution. We usually observe evolution from fossils because it's one of the easiest ways to. However, it's not the most conclusive way because there's mistakes or there's missing parts in the puzzle of fossil evidence. But fossils are remains or traces of past life, such as trails, footprints, or preserved droppings. And 99% of the organisms that we find as fossils are already all extinct. And they help show us a descent from a common ancestor. There are missing links, though. There are transitional fossils from a common ancestor for 2 different groups of organisms, and sometimes you can't find them. So you're confused at why there's these new 2 different species because you didn't find the link that connected the branching out from the 2 of them. So I want you to just kind of summarize overall how fossils are used as evolution for as evidence for evolution. So pause and go ahead and summarize that. We also can use anatomical evidence for evolution. I'll define these terms for you and give you some examples. So there's homologous structures, which are the same underlying structure, aka bones, but they have different functions. So example is bat wings and human arms, right? Bat wings are used for flying. Human arms are used for picking up things, kind of moving things around, grabbing things. And they both have bones, muscle, skin, and hair, but they have different jobs now. Analogous structures have different structures, but they have the same function. So it's kind of similar to earlier with convergent evolution, like I was talking about, where you have a bat wing and an insect wing. And the function is to fly, but the structures are very different because a bat wing is made of, like I said, bones, hair, skin, muscle, and insects are made of an exoskeleton, and sometimes they're made covered in scales as well. And lastly, vestigial structures are structures that serve no purpose currently to increase survival and reproduction, but they most likely did in the ancestor or the common ancestor. So over time, they're becoming less used. And so having a large functioning version of that organ or structure is no longer going to be the reason why an animal survives and reproduces. So smaller ones become more and more prevalent, and maybe slowly they will just disappear off the body of the organism. So examples are the human appendix. It used to be an important part of the digestive system for human ancestors, but we no longer need it. And sometimes you have to get it removed because sometimes if it gets infected, you get in really big trouble and you get really sick. Another one is the whale pelvis. The whale ancestor came from land, so they used to walk. But whales now swim and they don't need their pelvis bones anymore because they don't use or have any hind legs to be walking around with when they're swimming around in the ocean. So write 1 to 2 sentences explaining how anatomical similarities provide evidence for evolution. You can use the images to help you explain. Pause and go ahead. We also can study biogeography. So it is the study of range and distribution of plants and animals throughout the world. Biogeographical distributions show related forms of life evolved in one locale and then spread. So you can have a different mix of plants and animals existing in geography on different islands and continents and across oceans. And you want to think about Pangea too, because when the continents were shifting, what originally used to be a habitat and one landmass now has spread across and become very far apart, but they still have the same species on them. So maybe that's possibly why you're looking at some of our fossils from back then in very, 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 very separated locations. So go ahead and explain 1 to 2 sentences in summary of how biogeography provides evidence of evolution. You can also look at developmental similarities. So you can look at the fish, the salamander, the tortoise, the chicken, the human, and as they develop, they become more and more different. But at the initial stages, they look pretty similar because all the genes for development in our genetics that were the base genetics for the foundation of all life are all pretty similar. Like I said, we all have very similar genes for any living thing compared to each other. However, the way we express them ends up being differently. And the longer we express them for, and the more we express certain genes over others, will look more and more different from each other compared to other organisms. But initially, pretty similar, right? So write 1 to 2 sentences explaining how developmental similarities provide evidence for evolution. You can use these 2 as examples, but Google them quickly to explain, or at least know what they mean if you're going to use them. The last thing we're going to talk about is biochemical stuff. So that's DNA and protein, and we can look at it as evidence for sequencing them and looking at similarities or just differences because, like we said, most of our DNA is actually really, really similar with anything that's alive. So the reason we can do this is because all living things have the same DNA triplet code. So we have codons and we use ATGC in our DNA. And then when we use ATGC, we're going to end up making proteins, and it's the same 20 amino acids in proteins, just maybe different chains, different lengths of chains, different folding versions of chains, different combinations, but it's the same 20 amino acids. So when the DNA-based sequence is very similar between 2 species, it means that they have a recent common ancestor that they split apart from. Recent as in like maybe in the last 1 to 5 million years ago. And then when they're really, really, really different, that means that there was a more ancient common descent, or that common ancestor existed many, many years ago compared to, maybe 45 million years ago instead of just 5 million years ago. And it means that there was a common ancestor because there are some similarities, but there are also just more differences. So that's what we did in the BLAST lab. And here you can see we're looking at the dog and the chimpanzee, and you can compare them. And in the BLAST sequencing, you can actually see just how close they are to each other. And you can calculate out the differences and the similarities. And it's kind of cool because it's weird to think of how similar we are to all other living organisms. But in a sense, it makes sense because we all came from one common ancestor originally anyways. So go ahead and finish up your notes and make sure you turn them in for extra credit.